RNA editing is one step closer to the clinic. In our last post, we told you how current CRISPR advances allow us to not only edit the DNA, but also the RNA.
Many of you wrote to CRISPR asking us to tell you more about it, and almost with the perfect timing, HuidaGene released a paper about in vivo RNA editing in mice and non-human primates.
In this paper, which was published last week on Nature Neuroscience, the researchers used RNA editing to treat a rare condition called MECP2 duplication syndrome, or MDS.
This X-linked condition affects mostly male individuals, leading to intellectual disability, anxiety, motor dysfunction, epilepsy, and recurrent respiratory infections, with most patients dying by age of 25.
MDS is caused by a duplication of the MECP2 gene, leading to supraphysiological levels of the corresponding protein. Contrary to what we've told you so far, in this case the gene is working, but the increased amount of the MECP2 protein leads to this condition.
Unfortunately, not all conditions can be treated by knocking out a gene, and replacing a faulty gene with a fully functional one can be even more challenging.
Particularly in this case, the trick lies in maintaining a proper level of MECP2 expression, as too little or too much are both deleterious.
That's why HuidaGene decided to tackle this on a completely new angle. Instead of editing the DNA, they decided to target the RNA.
For this, they deployed their new HF-Cas13y. Like Cas9 or Cas12, this new editor can be directed towards its target with a guide RNA. However, it only recognizes RNA, not DNA.
The researchers first tested a cell model which guide RNA was better suited at knocking down the MECP2 RNA, obtaining up to 80% of regulation with a single guide and 90% with two guide RNAs.
And we know what you're thinking. Isn't that too much downregulation? Yes, but you see, this was just the first step. They wanted to select the most efficient guide RNAs. And after that, they moved to adjusting the dosage of the components to lower the level of MECP2 to that of a healthy individual.
With this in mind, they tested their approach by packaging the HF-Cas13 and the sgRNAs in an associated viral vector and injected it into the brain of mice with MDS.
After the treatment, they showed a normalization of the MECP2 protein levels and also a mitigation of the neurological effects caused by the condition, a huge success.
As the cherry on top, they also tested the approach in a large animal model, the non-human primate. This is the closest you can get before doing a test in a human patient.
Although they didn't have a non-human primate model with the MDS disease, they could still show successful downregulation of MECP2 using this strategy in a model as similar as possible to the human.
So there you have it, a real-world example of RNA editing. HuidaGene has paved the way for the application of RNA editing platforms in contexts where acting on DNA is not possible or even more harmful.
Their data also shows promises for the development of therapeutics against MECP2-related syndromes. When this happens, we here at CRISPR will be ready to tell you all about it.